Welcome back to Educator.com. This is the lesson on cells, parts and characteristics. So when we talk about cells as being the basic unit for life, you have to consider that microscopes are the key to really finding that out. And prior to the 1600s, cells weren't widely known. Cells weren't known to people as being the building block of life. You know, they would look at something and if it was moving and behaving like something they thought was living, well, they'd be like, oh, that's alive. But prior to the 1600s, looking at something like coral in an intertidal community or a sponge, they might think, that's not alive. It's not moving. But when you look closely, it's got cells in it. And those cells are definitely alive. So this was the key to advancements in cellular study. And if we trace kind of the major events in terms of microscope development and use in science, in the 1600s, Anton van Leeuwenhoek, he was very instrumental in terms of the first guy to actually use a simple compound microscope, having some lenses together and zooming in on something and seeing like, oh, I can see that little body, that little cell. He didn't call it that, but I can see that little unit. And now that I'm looking up close, it makes more sense how the whole exists. So this guy, very instrumental in terms of coming up with a cool way to zoom in and see the details. Also in that same century, Robert Hooke, an English scientist, he's the one who coined the term cell. And now it's just a common term, cells. But when you think about the reason why he came up with that, he was looking at cork. He was looking at basically the bark of these cork trees that you would find, in parts of Portugal, for instance. And if you slice cork very, very thin, you see what look like almost like little cells. And I'm saying cells with you knowing what that means, but when Robert Hooke was looking at these basically dead plant cells and just kind of looking at the shell of the cell wall of these plant cells, he thought like, oh my gosh, they look like little cells where a monk in a monastery would live, like a little tiny dormitory or room. And that's why he called them cells. So it just happened to be because he was looking at cork and looking at these dead, desiccated plant cells that we have that very common, important term now. You know, in the next couple hundred years, there were more and more advancements in terms of getting better and better microscopes, more lenses, compounding the magnification greater and greater. And then you get to the 1800s where these two scientists had big contributions in their own fields. Matthias Schleiden, a botanist, he's the one who really specialized in looking at plant cells and realizing what's true of plant cells. And Theodor Schwann, he's the animal cell guy. Schwann cells, actually, his name inspired the name for a kind of insulatory cell that's wrapped around the axons of neurons in animals. So this is the plant guy, and this is the animal guy. Flash forward decades and decades later to the next century. In the 1930s, you don't just have light microscopes anymore. So all of these scientists up here relied on light and lenses magnifying an image and bouncing light off of that so you can see what's reflected back at you. But if you want to get smaller and smaller and smaller and smaller, you get to the point where light is not as effective beyond 1,500 or 2,000 times magnification. When you get to 5,000 times, 10,000 times, 50,000 times magnified, you need to rely on electron microscopes. So in the 1930s/1940s, you start to have these, which are really good at figuring out, the shape of that molecule. What does the inside of a mitochondrion look like? A very important cell organelle. So these have to do with bouncing electrons off of a particular molecule, a particular item, and the way that they bounce off and they make an image, a very grainy image, but something that informs us about the shape of really, really tiny things. Today, there are two main kinds. There's scanning electron microscopes and transmission electron microscopes. In general, scanning electron microscopes give you a very good image in terms of like what a cell looks like as a whole, its shape and its structure. And transmission electron microscopes give you a really good idea of the way that parts are inside of cells in terms of their precise structure. The cell theory. This is made of three tenets, three important points that all have to be true for the cell theory to make sense as a whole. So the three tenets, these are agreed upon by all life scientists. In the future, it's possible it might be modified, but as it is now, this is the cell theory. The first tenet, all organisms are composed of one or more cells. So if you have something that is not made of cells, it is not alive. It has to be made of at least one cell, 2 cells, 16 cells, 100 trillion cells, and then we can call it an organism that's alive. The cell is the basic unit of structure and function for organisms, meaning every structure in a body, in a living being, and every function, in terms of every activity that organism is doing, it has to do with the cells inside of it. So the reason why your brain looks the way it does, the structure of your brain, comes down to the individual cells inside of it, known as mostly neurons. The function of your brain, every single activity, every single action your brain does, has to do with the cells inside of it. All cells come from pre-existing cells. That's pretty self-explanatory. You can't get a cell unless it's from another cell. And this is called biogenesis. Life creating life. The only example of abiogenesis, meaning life coming from no life, would be the very first cells on Earth. And that might seem contradictory to say, well, yeah, at one point in time, this wasn't true. But there are reasons for that. And in the lessons on evolution and the history of life, I'll bring that up in more detail. But yeah, the reason why you have your cells is they came from your parent cells. They have their cells because they came from their parent cells, and so on and so forth, all the way back to the first humans, and we can keep tracing back our ancestry to the very first life forms from about 3.5 billion years ago. Oh, and this picture down here, this is a zoom in. It's a little bit grainy, but this is a zoom in of blue-green algae, also known as cyanobacteria, and you can see the little chains of these individual cells. These are alive. When you're not zooming into it, it just makes, water look kind of greenish, a little bit murky. But there are billions and billions and billions and billions of these when you're looking at, a slightly greenish body of water in nature. The characteristics of life. So what is true of every living being? And these are generalizations, but these are true. Display organization. There's a certain kind of building in terms of how an organism is put together. Even something that looks amorphous at first glance, like this. This looks kind of like just a blob, but this is a paramecium. This is a living single-celled organism, unicellular if you want to call it that. This does display organization. It has a rim around the whole circumference. That's called a plasma membrane. If you look really carefully, there are tiny little hair-like structures jutting out of it. They're called cilia. Inside, there's a nucleus. There are lysosomes. There are ribosomes. There are all these structures that look organized in terms of kind of where they're at and how they behave with respect to one another. Grow and develop. Every organism has the ability to grow, to take on more matter and organize it as itself. And in terms of development, a lot of organisms start out as one cell, and as they grow, they develop tissues. They develop cells that have very particular functions. And this is definitely true of us. Reproduce. Yes, it's true that some organisms, humans for instance, can be sterile. They're unable to have children. But in terms of what our DNA is telling our species in general, we are meant to reproduce. We are meant to make more individuals, and life makes more life. So reproduction goes hand in hand with life. Responds to stimuli. It might not always be obvious, like, someone turning their head in response to light or a sound, but even this paramecium cell responds to stimuli, to activities or signals in the environment. There'll be a chemical signal that tells it, like, hey, there's food over there, or there's something coming next to me that could eat me, so it moves in the opposite direction. It's responding to chemical stimuli. Maintain homeostasis. Homeostasis means literally same state. And maintaining homeostasis is really the key to staying alive. We do this whether we're sweating or shivering. Sweating would be your body's response to increased body temperature. So sweating out some water can actually cool down your body. That's homeostasis. So your body doesn't overheat to the point where cells are getting damaged and there's permanent damage. Opposite with shivering. Your body temperature is a little too cold, so your brain makes your muscles contract and relax really quickly to generate heat energy. So those are both simple examples of how your body maintains a balance internally with respect to what's happening outside of it, and can evolve. Now this will make more sense with the evolution lesson, but an individual organism cannot evolve. I was born Homo sapiens sapiens. That's the species I'm a part of and subspecies. I will perish as that particular species. Populations over time can evolve. A population is a group of organisms of the same species in an area. But the ability to evolve goes hand in hand with life. Any cell has DNA in it if it's alive and functioning, and DNA over time can be changed gradually to give you some very different characteristics, and that's the key to evolution. Prokaryote versus eukaryote. If you look at all the cells that have ever been discovered or studied on Earth, they fit into one of these two categories. And I mean it. All cells can be included in one of these two categories. First one, prokaryote. The other term in terms of an adjective would be prokaryotic. So I could call a cell a prokaryote or say that is a prokaryotic cell. It means before the kernel, which sounds silly, but pro means before, and karyote comes from karyos, a Greek word that means kernel or nut. I had a student several years ago who was fluent in Greek, and she said that karyos is what I meant to say, can also mean walnut. And when she said that, something dawned on me that helped me remember what this actually means, and I'll draw it for you in a sec. But what this before the kernel has to do with is it means that these cells, they do not have a nucleus or other membrane-bound organelles. So the kernel is referring to the nucleus, this round body inside the cell. If you look at every single bacterial cell in nature, and all prokaryotes are bacteria, all bacteria are prokaryotes, none of them have a nucleus. So here's a bacterial cell. You'll have DNA in the center, and you'll have these things called ribosomes. These are not membrane-bound organelles. And you'll have a cell wall around the outside. But there will not be a round nucleus. There will not be things called Golgi Apparatus or Lysosomes, or these other things I'm going to teach you about later on in this lesson. So prokaryotes, no membrane-bound organelles, especially a nucleus. They evolved before the nucleus came into being, and they did just fine without a nucleus. On the other hand, eukaryotes is every other cell. This means true kernel or true walnut. It has a nucleus and other membrane-bound organelles. The example is really every other cell on Earth. Animal cells, plant cells, cells of different fungi. And then the one on the previous slide, that paramecium, that's a single-celled protist. It's also eukaryotic. So this whole walnut thing, the reason that has meaning to me is because if you crack open a walnut in just the right way, there's the shell rim. You kind of see this shape. Right? I mean, more or less. That's kind of the shape you see when you look at kind of the half of the nut that's in the walnut shell. And when that student, the one who spoke Greek, told me that, I was like, oh, it makes sense now. Because when you look at the nucleus of a eukaryote or eukaryotic cell, you see these. This is a duplicated chromosome, which you're going to see a lot more of later on in these lessons when we talk about DNA and cell division. If you use your imagination, you can see how this kind of looks like that, somewhat. And these are inside of a nucleus. So, this is kind of a basic prokaryotic cell, but here's kind of like we sliced in half an animal cell. Here's a nucleus. And inside you would see, it's from far away, but you would see these guys inside of there. And this is very typical of a eukaryotic cell. And actually there should be an even number if it's sexually reproducing, but that'll make more sense when we talk about meiosis. But you do see these chromosomes at certain periods of time within these eukaryotic cells, whether it's an animal cell, a plant cell, or fungi. And you also would see all kinds of other stuff. I'm doing a very quick drawing. You would see Golgi Apparatus, which looks like stacks of membranes. You would see things called lysosomes. And of course you would see ribosomes as well. Without ribosomes, and these are not membrane bound because they were found in here, without ribosomes a cell couldn't exist because ribosomes allow you to make proteins. So yeah, there's your basic designation between prokaryotic cells and eukaryotic cells. Here's a computer-generated image of the cell parts within a plant cell. So like I mentioned earlier with the cork cells that Robert Hooke looked at, you can see sort of that boxy look here with this green border. That is the cell wall. The yellow inside, if you look carefully, that's actually the plasma membrane, also called the cell membrane. Here's that nucleus, and certain times when the cell's actually dividing within here, the nucleus would actually break apart, and those little, those kind of like X-shaped looking chromosomes, some people call them like butterflies, you would see them. And this, of course, is a eukaryotic cell. Here's your nucleus. Here's all your other membrane-bound organelles. Anything that's a sac-like structure is membrane-bound. So here's your nucleus. We'll talk about this extensively in the next few slides. Golgi apparatus, vesicles coming from the Golgi, mitochondria, the key to getting a useful energy source inside of the cell, chloroplasts, how a plant actually makes sugars that are then going to be broken down in the mitochondria. This huge thing here, it looks like a reservoir of water. That's what it is. It's a water vacuole. Sometimes they call it a large central vacuole inside the cell. And then the cytoskeleton. There's other parts I'll tell you about in the next few slides. First one, let's start on the outside. The plasma membrane, also called the cell membrane, is the rim of every cell. The exception being if the cell has a cell wall, that would be on the outside. That would be superficial to this plasma membrane. But every single cell on Earth has a plasma membrane, and it's made up of the same basic components. It is protective and regulatory. Of course, it's kind of like a fence. It's kind of like that outer border, unless it has a cell wall. And it's going to be regulatory. Only certain things are going to be let in. Only certain things are going to be let out. If you let everything in and everything out, then the cell is going to be vulnerable to whatever might be in the outer environment. And it would lose things that are important to it. So for that reason, cells are known as semi-permeable. And what semi-permeable means is simply if it was just permeable in general, just completely permeable, anything will go back and forth. But semi-permeable tells us that like only certain things are let through based on what's needed or what's not needed. So that's the characteristic of every single functional cell. It's not going to let everything through. It's semi-permeable. Its membrane is semi-permeable. And it's composed of a bilayer of phospholipids, also just called a phospholipid bilayer. And here it is. If you were to take a cross-section, like slice through the cell and look at it, see what's inside of that little line that we see inside of like a microscope view. If you zoom in close, you can see the two layers. Here's the one layer, and here's the second layer. The lumen of the cell means like, hey, this is the space within a cell. A lot of times they call it the intracellular space. Here's the extracellular space outside of the cell. So in here, you would see the rest of the organelles, and here is an aqueous layer. You still would probably see fluid there as well. Notice that facing the fluid on both sides, you have these heads. And when they zoom in, they show you like, oh, here it is, okay? These are known as polar or hydrophilic heads. Remember earlier when we were talking about water, water is a polar molecule. So if this is a polar molecule, it gets along with water chemically or electrically. Polar, okay with water. So that's why both of these sets of hydrophilic heads are facing the water outside the cell and inside the cell. Hydrophilic literally meaning loving water. Philia is a Greek form of love, hydro, water. So hydrophilic, loving water. With nonpolar, which gets along with lipids, lipids are nonpolar, they don't like mixing with water. Also known as hydrophobic, afraid of water. That's the tails. So these tails, let me use purple for them. There are two. And these phospholipids actually look very similar to triglycerides, which were covered a little bit earlier in this course. The difference is that instead of it being a typical triglyceride, oh, like here's a glycerol and 3 fatty acid chains, there's just 2. And these are hydrocarbon or fatty acid tails. And taking the place of where that third one would be is actually a phosphate unit, which helps make up this hydrophilic head on this part and this part here. So you can see how they're oriented. There's this buffer region, a lipidy buffer on the inside made of these tails like this. But the heads are facing the inner aqueous region and the outer aqueous region. So this is a nice kind of like double border. It's a fence that's kind of like doubly effective in terms of it having this buffer and keeping the outside separate from the inside. Inside, there are proteins embedded. So what's not represented here is if, let me show you here with this. So there's a protein. They would call this an integral protein. It's actually spanning the length of this phospholipid bilayer. And this protein could serve several functions. It could be for transport, meaning it might have a little like hallway inside of it where stuff can go in, stuff can go out. It might be a gated protein. It only opens and closes when needed. It could be for identification. Your cells have very particular proteins in their plasma membranes that are slightly different from mine. We're the same species, but if I were to donate an organ to you, your body would be like, what? This doesn't belong here. And the reason why your body knows that is the proteins are slightly different. They would seem foreign. So sometimes proteins are just for identification purposes. Sometimes for attachment. Proteins allow cells to be bonded with other cells, attached to other cells. The reason why my skin doesn't tear apart here is I have proteins that are holding together my skin cells. And if those get damaged, then the tissue is going to fall apart. So a lot of times they are for attachment to neighboring cells or to neighboring layers. And sometimes they're enzymes. Sometimes there are enzymes embedded in the plasma membrane for the sake of changing a molecule, breaking it apart, building something, initiating a reaction. In general, this plasma membrane can be called the fluid mosaic model. That's a term for the fluidity of this. There have been studies suggesting that these little phospholipids, they don't just stay put. They are constantly moving around with respect to each other. So if you were to look down on this cell, it would look like a sea of moving parts. Those phosphate heads would be moving. It would look like an ocean. And they can move, not joking, like 1,000,000 times a second, which I can't even imagine, but there have been studies confirming that. The reason why it's called a fluid mosaic model is a mosaic means it has a lot of different pieces all together, like a tile mosaic, because it's not just the phosphate heads. You would see all kinds of different proteins scattered throughout this phospholipid bilayer. You would see what are called glycoproteins, little bits of cholesterol embedded in here to help with the buffer in terms of temperature and fluidity. So the fluid mosaic model is a term for how we understand the plasma membrane. And for the rest of this lesson, I'm going to compare the parts of a cell to a city, like an analogy. So like I mentioned before, this is like the fence on the outside of the city. So this will help keep it straight in terms of all the different parts of the cell and how they influence one another. Next is the nucleus. Now, if we're going to maintain this city analogy, the nucleus would be very much like City Hall. That's what's running the city, making the rules, making sure people understand how that works. And the nucleus very much is like the brain of the cell. I'll come back to that in a sec. So the nucleus, this spherical structure, it's got a double membrane made of phospholipid bilayers, like I mentioned before. It's also in here. It's not just in the plasma membrane. Inside of it, it's got the DNA, deoxyribonucleic acid, in a nuclear envelope. So notice this term here, nuclear envelope, and they say outer membrane and inner membrane because, like I said, it is a double layer of those phospholipid bilayers. So the nuclear envelope, there it is. Inside, you've got something called chromatin. So they're labeling it here, chromatin. You've got heterochromatin and euchromatin. Don't worry about the difference, but basically chromatin is all of the genetic information, the DNA, and associated proteins that are helping organize it. And those proteins are typically called histones. When we talk about how DNA is arranged inside of a chromosome in future lessons, you'll see histones specifically and how DNA is kind of wrapped around those histones. So chromatin is all that stuff on the inside. Nuclear pores permit the passage of molecules in and out. So if you look carefully, they're labeling the nuclear pores. Right there, right there, right there, right there, right there, right there. Stuff goes in, stuff goes out. You sometimes will have hormones that have signaled a cell like, hey, we need a certain protein to be made right now. And that hormone will go all the way inside the nucleus and affect the reading of the DNA and making of a signal to go out of the nucleus. Constantly, the nuclei, which is plural for nucleus, let me write that down. The nuclei of your cells, if the cells are metabolically active, they're constantly making RNA. It's kind of like a photocopy of DNA that tells a cell how to make protein. And yeah, it's very much like the brain of the cell. Like City Hall is in charge of a city. It contains the codes for how to make every single protein in the cell, how to make every cell part, how to maintain it, and how to keep the cell alive. It also has something called a nucleolus. So here is the nucleolus, which is like a mini nucleus in a sense. It makes ribosomes. That's the purpose of it. So ribosomes, which are actually labeled in this drawing, these little pink dots. Up close they look a little bit different, but in cell diagrams they look just like dots from far away. These are little protein factories, and without the nucleus or nucleolus specifically making these, a cell wouldn't be able to stay alive. So that's a summary on the nucleus. Next up, ribosome. Ribosomes are found in every single cell on Earth. Now, a nucleus wouldn't be, because if you remember, a prokaryote does not have a nucleus. It still has DNA, but it's not in a spherical structure inside of the cell. But every single cell on Earth, prokaryote, eukaryote, has ribosomes. These are not membrane-bound. They do make proteins. That's what they do. So here you've got two subunits. They actually have this unit of measurement here, 200 angstroms for the actual length of this, which is very, very tiny. This right here is a large subunit. This is a small subunit. A lot of times it's depicted like this, more simply. So if you use your imagination and tilt this on its side, you can see how they have this. This is looking from that angle, like at the bottom. But you've got the large subunit, small subunit. And it's got inside of it RNA and protein woven in a very particular way to give you this sort of, form, this globule kind of form. And inside of here, you have another kind of RNA that will move through. It's called messenger RNA. And then these things called tRNAs come down in here and assemble an amino acid chain. That's how they actually physically make proteins. More about that in a future lesson. They're found throughout the cytoplasm. This is a term for all that fluid area inside of a cell that's bound by the plasma membrane. They can be free or bound. What that means is a free ribosome, it can be floating around anywhere. It can be, next to the plasma membrane. It can be very close to the nucleus, just kind of floating around, reading RNA. Also, they can be bound. They physically will dock on what's called the ER, the Endoplasmic Reticulum, which you're going to hear about next. They use various kinds of RNA to assemble proteins. I mentioned those a moment ago. mRNA, tRNA. Inside of it, there's ribosomal RNA, also known as rRNA. And amazingly, there can be 1 million of them in one cell, which is really hard to picture, but these are little factories that are making a very important product for the cell. So to maintain that little city analogy, these are factories. Little shops. You could think of it as a shop that produces a certain product, and that product is protein. The protein is like the major export. It's the thing that the city relies on to stay functioning and to stay alive. Next up, the endoplasmic reticulum, which in Latin literally means little net inside the cell. It kind of does look like a net if you use your imagination. It's also known as the ER for short, not emergency room, endoplasmic reticulum. It's typically adjacent to the nucleus, like right outside of the nucleus. And if you look very carefully at this animal cell here, there's the nucleus and there's the ER, which they're zooming into here. So if this was an ER, the nucleus would be right there. And it does look like a membranous hallway, like you can imagine different molecules kind of weaving through here as it goes through the different membranes. And it's a site of numerous chemical reactions. Depending on where you are in this ER, you've got different stuff going on. Sometimes it's proteins being made, lipid parts being made. Ribosomes are doing a lot of work in this area. You can see all those little yellow dots. Those are ribosomes. And the ER can be rough or smooth. The rough part looks like if you were to touch it, it almost looks like it'd feel like sandpaper. It'd be rough because there are ribosomes. This looks kind of smooth. So here there are no ribosomes. That's why it's a smooth ER. So like I said, rough has ribosomes, mainly for protein synthesis. Because if you remember from the previous slide, ribosomes, they're those little protein making factories or shops. And so if you've got a bunch of these protein makers on here, you would expect that there'd be a lot of protein making. And the reason why it's important they're on this membrane is once the protein is made, it can be wrapped up in a little membrane phospholipid bilayer unit. It's actually the same membranous structure you see in the plasma membrane. And that little membrane sac can take the protein to the next organelle, which you're going to hear about in a sec. The smooth ER right here. Depending on the cell, you would have more smooth ER than rough ER. There's not always majority rough. Something like a liver cell would have a lot of smooth ER because of the making of lipid products in that particular organ, and also the breakdown of certain chemicals, certain toxins. You would need a more extensive smooth ER. There are no ribosomes here. And it's mainly for lipid synthesis and storage, the making of fats and the secreting of fats for the health of the cell. So that's the ER. To maintain that city analogy, this would be, I'm going to call it a special cart or you could think of it as a pathway. But imagine that when these products are made at the factory, there's a special cart that ships them to where they need to go within the city. So this special cart allows those products, those proteins, to go out to where they need to go to. Next up, the Golgi Apparatus, also known as the Golgi Complex, the Golgi Body, depending on what source you look it up in. But Golgi Apparatus is a very common term for it, named after an Italian scientist, Golgi. He discovered it. It looks like flattened membranous sacs. So oftentimes you'll see it like this. A cross section I've seen before kind of looks more like this. Looks very similar to this image. And you'll see little, they call them secretory vesicles. Little sacs will kind of pinch off of them, similar to what I was talking about with the ER, how a protein could be kind of trapped or contained within a membrane. You'll have secretory vesicles going to the Golgi and leaving the Golgi, and here's why. The Golgi packages, finalizes, and transports proteins that have come from that ER. So typically you'll see the Golgi neighboring the ER in a cell. So imagine this is like a postal office or post office. So that special cart takes that protein product to a post office where it's, packaged, tagged with a certain zip code, a chemical zip code attached on that particular protein, so that it goes to the final destination where it needs to be. So membranous vesicles, which is just a term for like container membrane, these vesicles will fuse with it that have come from the ER containing an amino acid chain or polypeptide, also known as a protein product, and they go through the layers. That protein gets modified, put together with other proteins. Like I said, little chemical sugary molecules can be attached to it to give it kind of like a zip code where it needs to be. And then it pinches off and ends up going to the destination. And that's a summary of how it happens. They go in one end, they come out another end. And that's the Golgi Apparatus, also known as the cell's post office. Okay, we're going to review what we've gone over so far. We have plenty of others to talk about, but I just want to show you spatially how these things are related. So we started with the plasma membrane, all of this yellow wrapped around, made of that phospholipid bilayer. Well, guess what? The nucleus also has phospholipids in its envelope. The ER, phospholipids. The Golgi, made of phospholipid membranes. Coincidence that they all have the same? Actually, no, it's not a coincidence. It's meant to be that way. This is known as the Endomembrane System, and I haven't covered all of the parts of the endomembrane system. Another one is the lysosome, which is around here somewhere. Where are the lysosomes? Oh, well, peroxisome is similar to a lysosome. That could be a lysosome right here in blue. Any of these little sacs or membranous parts tend to be part of the endomembrane system. There are some exceptions. But what that means is their little phospholipidy layers can fuse and interchange with one another, and that is for convenience. Imagine that you have instructions coming out of the nucleus going through the ER, you wrap it up in a little sac, and if that sac is not compatible, that vesicle is not compatible with the Golgi, that'd just be more arduous for it to work out. So that sac can easily fuse with the Golgi. Those parts just can go together quite simply. And then when that product leaves, these little Golgi vesicles, if they need to go out of the cell, maybe they have a little sac of hormones that need to be sent out into the bloodstream, well, that sac can easily fuse and dump out its little the parts it was containing, and that's called exocytosis. And it can actually dump those things out. And thanks to the endomembrane system, those are compatible. Parts that are not compatible, the chloroplast and the mitochondria, they are standalone. And you'll see in future slides in this lesson, they do not easily fuse with those things. A vacuole is another example that can fuse with this. So a vacuole, its border has that same kind of membrane, and things can fuse with a vacuole or a lysosome, or even peroxisomes. Ribosomes definitely are not a part of it because remember, ribosomes are not membrane bound. They're like two little chunks that fit together like this. Moving on, mitochondrion. Plural, mitochondria. So don't say mitochondrions. That's not a word. So mitochondria, plural. The majority of your cells that have mitochondria would have many, many, many of them. And these are membrane-bound organelles, not part of the endomembrane system though. And they have their own DNA in ribosomes. That's the amazing thing, that inside of here, there actually is mitochondrial DNA. And ribosomes that are unique to this mitochondrion. And with both this and chloroplasts coming up next, there's a lot of evidence suggesting that this is the descendant of a previously independent cell that a long time ago, billions of years ago, ended up inside of a larger cell. And it worked out, it persisted. And when that cell divided, the mitochondria divided and stayed with it. And we have mitochondria. Majority of plants have mitochondria. Fungi do. So mitochondria, very important in terms of producing energy for the cell. It has an outer double membrane right here. It also has, phospholipids, but the structure of it and the proteins in there are not compatible with the endomembrane system I mentioned before. Inside, there's a highly folded inner membrane called the cristae. So all of this yellow, you can see it is kind of weaving. That is highly folded and compacted in there. And the reason why it's highly folded is important. You have a lot more membrane in here when it's folded up, just like how the outside of your brain is folded. Those convolutions of your brain can jam-pack a lot more matter into that area when it's folded. It's the same idea here. You have a lot more chemical reactions happening inside of the mitochondria thanks to the cristae that's folded up. And what does it do? Well, it's responsible for producing energy-storing molecules known as ATP, adenosine triphosphate. This will come up a lot more in the unit on cellular respiration and in photosynthesis. This is kind of like energy currency for the cell. You take in food, you and I, we are known as heterotrophs. We have to take food in as an energy source from outside of ourselves. We can't make our own food. Well, then what do you do with those sugars, those lipids, those proteins to give your cells energy? You can't just take a sugar and attach it to a part of a cell and be like, move. No, that sugar needs to be broken down and kind of transferred into this form known as ATP, which can easily be spent. It's, like I said, it's like energy cash for the cell. You can spend it on doing anything in the cell that requires energy. That's what this wonderful structure does. It helps break down sugars, organic compounds, to give you this energy currency. And this structure is very much kind of like, I would call it a hydraulic dam. This is what energizes the city. A hydraulic dam has a lot to do with water movement that ends up fueling the electricity source. But in here, the dam is not that far off, because you'll see when you look at the lessons on cellular respiration, there are these enzymes embedded in the cristae called ATP synthase. It's actually an enzyme that makes this. And as charged particles, specifically protons, move through that ATP synthase, it spins, and it's kind of like water moving through a water wheel, except it's charged particles spinning an enzyme that helps make this. So the mitochondria are very much the power source for the cell city. Chloroplasts, found in plant cells. You would not find these in animal cells. I've heard of one animal that's ever been discovered. It is a kind of sea slug that does have chlorophyll inside of its cells, which is a pigment molecule that allows it to absorb sunlight, and its body's kind of shaped like a leaf. But it still has to eat. And it doesn't have chloroplasts. It does make chlorophyll in little bits, but I stand by my point that these are only found in plant cells, not in animal cells. They are membrane-bound organelles with their own DNA and ribosomes. That should sound familiar because we just said that with mitochondria on the previous slide. Yep, inside of here there's DNA and the ability to make ribosomes. And that's important because, like in the mitochondria, there's a lot metabolically going on in this structure, and it kind of regulates itself. Just like in the mitochondria, there's an outer double membrane, and there's folded membranes on the inside, but for a different purpose. These are not called cristae. These they look like green pancakes, are called thylakoids. Let me highlight that in green. Thylakoids. And one stack of them, you can see the label here, is a granum. So there's a granum, 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 granum. Plural would be grana. So you can see if you look at these stacks, we have plenty of grana inside of the chloroplast. The area outside of the grana, labeled here, is called the stroma. So right now I'm tracing over where the stroma is, the fluid outside of the fluid-filled thylakoids. And the parts of the chloroplast do correspond to the different phases of photosynthesis. There's a whole lesson on that if you look in the future lessons in this class. The purpose of the chloroplast, it's responsible for producing sugars through photosynthesis, like I said. So plants, autotrophic, meaning they make their own food, they provide their own food energy. So a plant makes sugars thanks to this, and then it would send out the glucoses, the sugars, to be broken down in a mitochondrion to give the cell ATP. So plant cells typically have mitochondria as well. This is very much like the solar plant for the city. And in cell city analogies that I've seen before, they will sometimes leave out the chloroplast because they're taking it from the point of view of an animal cell. An animal cell doesn't have a chloroplast, but if you want to have some image of what this does inside of a cell or what it could do for a city, it's very much like the solar plant. Vacuoles/vesicles. I've included these in the same slide because they structurally look very similar. A vacuole is a temporary storage container, and a vesicle has something contained in it, and it's typically going somewhere. It's going from the ER to the Golgi, or from the Golgi to the plasma membrane. The difference in these two is really what they contain. So they are both membranous sacs for storage or transport. Vacuoles can contain food or water. Now, typically in a plant cell, you would see water vacuoles. We saw that in the image earlier in this lesson. It looked like a big lake. These food vacuoles you can also see in plant cells. So once they've made a bunch of sugars through photosynthesis in that chloroplast, they will store them away. One place is called an amyloplast, and that would be a place for them to store starch. But the storage of food, whether it's in an animal cell or a plant cell, they would call it a food vacuole. Like I said earlier, in plants, they tend to be large central water vacuoles, looking like a big lake, as you see down here. Now with the vesicles, structurally the same border in terms of that little phospholipid sac. It's for transport of cellular products or wastes. So a vesicle can go from the ER to the Golgi, bringing a protein to the Golgi to get modified, packaged, transported. From the Golgi to the plasma membrane, like I said earlier, it could contain hormones that need to be dumped out, and that vesicle will fuse with the plasma membrane. It might have wastes. Maybe invading viruses were eaten up by a part of the cell, which we're going to hear about in a sec. And then a vesicle fuses with it and takes those broken up pieces of the virus, which is now waste, to dump outside the cell. That can also happen. This is a connection of the water-filled vacuole that you would see in a plant cell, and these terms having to do with osmosis and the concentration of ions or salts in and out of the cell. So this is basically explaining how you would get a plant cell getting really puffy, getting filled with water in that vacuole, or losing some water and getting kind of shriveled up. These terms will come up a lot more when we talk about osmosis with transport in and out of cells. But you can see that this is known as turgid, meaning it's quite puffy. Water ended up pouring inside of this because it was more highly concentrated with salt and other ions, and that drew in the water to fill in this big vacuole, and now you have what's called turgor pressure, coming from this word. Turgor pressure, that's pushing on the plasma membrane, which is then pushing on the cell wall. The opposite's happening here. There was so much water that left that you can see that the cell just kind of just desiccated and shriveled. So much water left out of that large central vacuole. In terms of the cell city analogy, I mean, these are just like storage containers. Or transporting containers for the products and the different items that are associated with those factories or shops. Lysosome. Lysosome comes from the words lys and some, which means a body that breaks down stuff. So lys, the word lys, means to break down, to split apart. And that's what these do. These are little membranous sacs that have enzymes inside of them that are meant for breaking things down. Could be the breakdown of macromolecules. Maybe the plant cell or the animal cell has stored a polysaccharide that needs to be broken apart into little bits of sugars to be made into ATP, that energy molecule. So the breakdown of those macromolecules can happen a lot quicker if you have lysosomes that are doing that. The breakdown of damaged cellular parts. Maybe the Golgi apparatus is really beat up and damaged, and a lysosome will go over and sort of eat up its little parts, help break it down, and then those parts could be recycled to rebuild the Golgi apparatus. Breakdown of foreign bodies and invaders, whether it's a virus, bacteria, fungal body. If it's entered the cell and has been identified as foreign, that it doesn't belong there, the lysosomes can be stimulated to fuse with it, kind of like eat it up, and the little enzymes inside of it will help break down those molecules. You notice that some of these little enzymes are represented with happy faces. Very, very happy for the cell, but not happy for the virus or bacteria that it's eating up. And yeah, they contain digestive enzymes, typically. They're meant to break stuff down. They're not meant to just end up all over the place in the cell. And I say that because it's very important that they say it contained in the membranes. I've heard that if every lysosome inside of a cell were to burst simultaneously, it would cause autolysis. Autolysis meaning self-breakdown. It would literally destroy the cell. Another term is apoptosis. If that was stimulated to happen like it's supposed to happen inside of a cell, that's programmed cell death, apoptosis, and as a result of autolysis. So if all of these simultaneously were to leak, it'd break up all the parts of the cell. So how does this relate to the city? A lysosome would be waste disposal area, garbage dump, whatever you want to call it, recycling plant. There's a lot of terms you could give to this related to how a city works. Centrioles. Centrioles, found in pairs, not typically in plants. It's made of a cylindrical ring of microtubules, and if you look very carefully here, you can see that. They're both cylindrical. You can see how they're kind of perpendicular to one another. Very carefully, if you look here, you've got these little yellow rods. Each of these is a microtubule, which is a protein rod, basically. And you can see there are 9 groups of them kind of lined up in the cylindrical form. They are contained within centrosomes. So these two would be in a region that is known as the centrosome. 2 centrioles and a centrosome. And they have an important function in cell division. When you see a cell dividing, and I'll give you a very brief image of what's known as metaphase. Here's the centriole pairs. The centriole pairs, they get duplicated before a cell is going to divide, and they end up at the poles. So here's the plasma membrane. centrioles, and lined up down the center, this is after the nucleus will break down, these are 4 duplicated chromosomes. And what I'm drawing here is microtubules, besides the microtubules in the centrioles. These are kind of like little protein ropes that are anchored by the centrioles here and the centrioles here. So you can kind of see how they're like kind of holding down these threads. And what's going to happen next is these microtubules will get shortened. And as they get shortened, they pull on these little X's and split them in half so that you get the duplicated chromosomes or pieces of DNA moving to what are going to become 2 different cells eventually. So that's a very rough drawing, but showing you how these participate in cell division, and they're anchors for the spindle apparatus. This term is describing what I drew here. This is the spindle apparatus that allows those chromosomes to be pulled apart. In terms of the cell city analogy, I don't have a representation here for centrioles. Just know that they really serve an important function when it comes to splitting apart chromosomes, particularly in animal cells. The cytoskeleton, I just mentioned a part of the cytoskeleton in the previous slide when I said microtubules. So the cytoskeleton forms a framework or scaffolding for the cell. It kind of holds it together and prevents the cell from sort of collapsing in this sort of fluidy blob. So the cytoskeleton is very much like the skeleton of the cell, and those parts are kind of pushing on the inside of the plasma membrane. It provides a network of protein fibers for travel as well. There are really cool videos out there on the internet that show a motor protein walking along a microtubule, transporting a transport vesicle to the surface of the plasma membrane. And that motor protein, it looks like it's alive, but it's not. It's much smaller than a smell. I just said smell. I meant it's much smaller than a cell. And the motor protein is walking because ATP molecules are being attached and broken apart and attached and broken apart to shift it. And it does this little walk along the microtubule because these protein fibers can kind of serve as like little pathways or roads throughout the cell to get from one organelle to another. This cytoskeleton is made of major roads, some smaller roads, and then the little tiny roads that connect houses in a neighborhood. So the major highways or the thickest units are microtubules. So these are the thickest of these protein rods by far. This is how a microtubule is built. It is a helical arrangement of tightly wound tubulin protein units. So these are all little amino acids strung together to make up tubulin. And if you cut a microtubule and look at it, this is what you would see. And you see there's alpha tubulin and beta tubulin, just 2 slightly different forms. Intermediate filaments. These are more thin compared to the microtubules. The way I remember it is intermediate, like in the middle. It's literally in the middle of this one and that one. Microfilaments, not made of tubulin, they're actually made of actin, typically, just a different protein. These are the thinnest by far. These little microfilaments, don't think they're useless. They're actually very important when a cell like an amoeba, amoebas tend to be like, they look kind of amorphous, and you would see ribosomes and all kinds of vesicles throughout. But the way that amoebas form their pseudopodia, it looks almost like these kind of fake feet, like they're crawling through the water. The way that they make these little sort of undulations of their plasma membrane is they change the way that microfilaments are pulling or letting go on the inside of the plasma membrane. And that all comes down to using ATP, that energy molecule, to manipulate the shape of the cytoskeleton. But that's how they literally kind of crawl through water, is their cytoskeleton. Cilia. Cilia are like little cell hairs. The singular word would be cilium. So this is plural. So I wrote the plural word for this slide because generally on cells, when you see them, you're going to see a lot of them. They are short, numerous projections that look like cellular hairs. And this is a micrograph image. It's zoomed in thousands of times. You're looking at the surface of human cells. Chances are this is in the bronchial lining. You have a lot of cilia in your respiratory tree that help sweep up mucus. So, like the centriole units, it's made up of a ring of microtubules. A little bit different structurally, but each one of these little hair-like structures that's projecting out of the plasma membrane. And if you look carefully, I don't know about you, but I can see little dots here. All those little dots in this drawing, those are phospholipid heads. So those phosphate heads, part of that phospholipid unit. So yeah, they are sticking out through the plasma membrane. And if you do cut them in half, there's a ring of microtubules. They move back and forth to make the entire cell move. So it's almost like oars on a rowboat. They move a lot like oars, back and forth, as if you are rowing with a rowboat. And I say that because the next slide has a different structure that doesn't move in that way. These move back and forth. So sometimes, if we're talking about a paramecium, and I'm not going to draw all of them, but a paramecium is a single-celled organism that would have tons of cilia all along the outside of it. And these cilia move back and forth in a timed fashion to make this cell move wherever it wants to go inside of a lake or a pond or whatever it might be. They move very fast. So it's impressive when you look at them alive under a microscope, how quickly they can move thanks to just these cilia. Also, in our body, it's not moving the cell as a unit, as a whole. Really, our cilia inside of our bronchial tree especially is to move substances outside of the cell. So imagine that this is not a paramecium, that this is just a cell embedded in your bronchial tree lining. So imagine that this is a section of the bronchial tree. And here's where air moves, in and out. And you've got all these little cilia that are projecting into what's called the lumen, or the passageway of your bronchial tree. As mucus is generated, you don't want the mucus to just end up pooling in your lungs and you're suffocating on your own fluids. You want to be able to sweep that up and out. And without these cilia, you'd be more likely to drown in your own fluids, get something like pneumonia. So these help keep us alive, the cilia inside of our bodies. Flagellum. This is another structure for movement, very different from cilia. There are some commonalities, but plural for this, you wouldn't say flagellums, you would say flagella. So if a cell had 3 tails, well, look at those 3 flagella. It's long compared to the cilia, much longer, and it's like a tail-like projection coming out of the plasma membrane, made of a ring of microtubules, just like cilia. So if you were to take this sperm and cut through the flagellum and look down it, you would actually see the same basic components looking down. It's just that the length of it and the movement of it is very different. Instead of moving back and forth like an oar, these move in a whip-like motion to propel a cell through fluid. So imagine that my arm is a flagellum. Okay, and it's anchored in the plasma membrane, like, my chest and back area is the surface of the plasma membrane. This doesn't move back and forth like that. No. That would be cilia. This moves kind of like you're turning a wheel back and forth. And as I turn the wheel back and forth, look what happens. That's how a flagellum moves. That's what would propel a sperm trying to find the egg in a female. So the only human cell that should have flagella is sperm. Sorry, ladies, you don't make cells with flagella. But you would actually not only see these in animals, there are some plants that actually do have flagella on their gametes, plant sperm. I know that sounds weird. They're structurally different from animal sperm, but 2 examples, ferns and moss have actually been known to make sperm. And those little cells need a fluid medium to move through and reach the egg. So cells typically have only one or two. A male human sperm is supposed to have just one tail, but they could come out with two. They could come out with more. More have been discovered on some cells. I've heard of up to 8. At a certain point, it gets pretty crowded. And I mean, how much better is 8 compared to 6 compared to 4? So sometimes there's too many of these flagella because of a mistake. Like I said, human sperm are supposed to have one. There are other cells in nature, dinoflagellates, a single-celled protist in the ocean, have many flagella, and it actually helps them kind of spin in different ways and orient themselves different ways inside of the ocean. Cell wall. It's on the outside of the plasma membrane, not in animal cells. You would never see a cell wall in animal cells. They tend to be more kind of rounded in shape. It really does depend on which cell. Something like a neuron would be very long. But plant cells like this image here, this computer-generated image, is very boxy thanks to the cell wall. Fungal cells have their own cell walls. Bacterial cells have cell walls made of different material. So it's extra protection and a rigidity for the cell. It helps them keep a certain shape. Think about this. Plants don't have a skeleton. We have a skeleton as a vertebrate, as an animal that walks around on land, and we have our posture and ability to move around. A plant, helping to keep its kind of rigidity to rise up, reach the sun, be competitive with other plant cells, or sorry, plant species is what I meant. You want it to have some kind of rigidity with the cells being stacked upon each other. So the plant cell wall, not just for protection, but also rigidity. And then you get a bunch of water pressure, especially inside of that vacuole, that large central vacuole, and that can help push on the inside of the plant cell wall and provide that turgor pressure. There actually is a 2-layer wall, and there are these little pores called plasmodesmata that happen to be connecting them. I think that's what they meant to draw here. Plasmodesma is one, and plural would be plasmodesmata. Those are the pores inside of the cell wall connecting plant cells to each other. So in plants, all this green stuff is made of cellulose. That is a polysaccharide made of glucoses linked together. Helps give it a good structure. In bacteria, usually, but not all species, you tend to see peptidoglycan making up a bacterial cell wall. Part protein-based, part sugar-based. That's how you get the peptidoglycan. And then in fungi, a different polysaccharide, they're made of chitin. This is that same polysaccharide that's in insect exoskeletons or the exoskeletons of crustaceans. One of the many pieces of evidence connecting animals and fungi in the whole evolutionary tree. But in fungi, yeah, a different polysaccharide than cellulose, chitin cell walls for mushrooms and the like. And then cytoplasm. I'm just going to draw a brief picture here. There's a cell, nucleus. So of course this is eukaryotic. There we go. It looks three-dimensional enough. So the cytoplasm is the fluid-filled region of a cell between the nucleus and the inside of the plasma membrane. So imagine that this is our phospholipid bilayer. Here's our nuclear envelope. From here to here, all of that is called the cytoplasm, all of this. So the majority of the organelles are in the cytoplasm. And it's a site for the majority of the cellular reactions. Not a surprise. The inside of the nucleus is not called the cytoplasm. I've seen the word nucleoplasm or nuclear fluid. But yeah, the cytoplasm is all of this. It's also referred to as the cytosol when they're talking about the fluid itself that is in the cytoplasm, cytosol. And finally, just to review some similarities and differences between animal cells and plant cells. So here's an image of an animal cell and a plant cell. You've seen this several times in this lesson. This is the first time you're seeing this. So let's start kind of in the order we went. These are the organelles you would see in both. You would see the nucleolus, the nucleus. You would see the ER, both rough and smooth. Number 8 is the smooth ER. 4, that would be a little vesicle that is pinched off of the ER going to the Golgi. You would definitely see that in plants as well. Number 6, there's the Golgi, right? You need to have the Golgi to finalize, package, and transport those proteins. Ribosomes. You certainly would see ribosomes in plant cells, all those black dots. Mitochondria, number 9. Yep, you definitely see them. Right here, they're a different color in this image. So here's mitochondria. Mitochondria. There's no centrioles in this picture. If you look carefully, there aren't any. Here are the centrioles. So this is one example of something you would see in an animal cell that tends to not be in a plant cell. Lysosomes, number 12. You sometimes do see lysosomes in plant cells. So I'm just going to assume that that blue dot and that blue dot there, that little sac, is a lysosome. So in some plant cells, lysosomes have been discovered. They're not in all plant cells. Number 10 could just be food vacuoles. And you definitely would see a water vacuole here. So vacuoles can be found in both. Plasma membrane, we don't want to leave that out. 14. Yep. That's the yellow right here. That's the plasma membrane. Number 7 is showing you a very small part of the cytoskeleton. It looks like a little thread. So remember, that's microtubules, intermediate filaments, microfilaments making up a scaffolding inside the cell. You definitely see that in here too. You can see little bits of the cytoskeleton in the plant cell. Now I'm going to circle some things in the plant cell that are not over here. Of course, chloroplasts. You would not see chloroplasts. Here's another chloroplast, but this one we're looking inside of it. You're not going to see chloroplasts inside of animal cells. Animal cells require the intake of food. They're not making their own food like a plant cell. And of course, the plant cell wall. You don't see that boxy outer cellulose wall here. You see a very kind of rounded out, typical looking animal cell structure. So that's a reminder of a lot of these parts inside of the cells and general differences between animal cells versus plant cells. Thanks for watching Educator.com.